This is a video about the fifth required practical activity in GCSE Biology and Combined Science, the investigation of the impact of pH on the activity of the amylase enzyme. By the end of this video, you should know what iodine solution is, and you should be able to explain how it can be used to determine the rate of reaction for amylase. You should be able to describe how to set up the amylase required practical and also how to interpret the results of that practical. Firstly, let's recap what we mean by amylase. Amylase is one of the many thousands of enzymes that are found in your cells, and an enzyme means that it's a biological catalyst. In other words, it's a molecule that speeds up the rate of chemical reactions without being used up itself, and it's made by a living organism, hence the biological bit. It isn't alive itself, and it's made from protein, so cells that make a lot of enzymes often have a lot of ribosomes. Now, enzymes have lots of different jobs, including making things as well as breaking things down. Amylase is a catabolic enzyme, so it breaks down another molecule called its substrate. In this case, the substrate is starch, and the starch is broken down to make sugar, not glucose. That's a really common mistake. It's actually a different sugar called maltose, but you don't get to hear about that one until A-levels, so for now, sugar will do. You should also note that lock and key theory tells us that enzymes are specific. In other words, they have a highly folded and unique active site, which interacts with their substrate because the shape is perfectly complementary, like a hand and a glove, or indeed a lock and a key. Enzymes can be denatured, which means that their active site is permanently damaged and bent out of shape, so that it can no longer interact with the substrate, and this will stop the enzyme from being able to work anymore. This can happen if the enzyme is exposed to extremes of either temperature or pH, and this is what we're going to investigate in this required practical. Before we go any further, pause the video and make sure that you can answer these three questions. An enzyme is a biological catalyst, and it's also made from protein. The substrate for amylase is starch, and when starch is broken down by amylase, it makes sugars. Crucially for this required practical, we're going to need a chemical called iodine solution. Iodine solution on its own is a dark orangey brown colour, but in the presence of starch, it turns a dark blue-black. And so this means that we can use it to detect whether amylase has successfully broken down starch into sugars. If we add a solution and the iodine stays orange-brown, then that shows that no starch is present, and therefore if we added starch in the first place, that means that amylase has done its job and broken that down. If, on the other hand, the iodine turns blue-black, that means that the starch is still present. So that could be because the amylase is still working and it hasn't finished digestion yet. It could be that we didn't add any amylase in the first place, or it could be that that amylase has been denatured. So its active site has changed shape and it's no longer able to break down the starch. So before we look at the practical, pause the video again and make sure that you can answer these three questions. We test for starch using iodine solution, and if there's no starch present, then this is a dark orange-brown. However, if starch is present and we have a positive test, then the iodine will turn blue-black. In required practical activity 5, we investigate the effect of pH on the rate of reaction of the amylase enzyme. This must be done using a continuous monitoring method, which means we're going back to our reaction again and again at regular intervals, usually about every 30 seconds. Also, we specifically have to control the temperature of the reaction using a water bath. Now, as with all of your required practicals, you need to be able to identify for this one what the independent variable, the dependent variable, and the control variables are. So the independent variable is the thing that we are changing in this investigation. When we do it again, what's different? So in this instance, it's going to be the pH. We're going to repeat the same reaction several times using several different pHs and see which one works best for the enzyme. Then the dependent variable is the thing that we're observing and measuring and seeing how is it affected by the independent variable. So in this instance, it's going to be the rate of reaction of the amylase enzyme, which here we're going to work out by looking at the length of time for all of the starch to be digested. And then the control variables are the things that we keep the same between the different tests in order to make sure that the data is valid, because if we changed more than one thing at once, we wouldn't know what was causing the change in the rate of reaction of the amylase enzyme. So we're going to keep both the concentration and the volume of both the starch solution and the amylase solution the same, and we're also going to control the temperature of the reaction using a water bath. It's really important that wherever you're identifying temperature as a control variable, you're actually doing something to control it. Saying that you'll do the reaction at room temperature is not a valid way of controlling the temperature. Before beginning this experiment, we prep a spotting tile with individual droplets of iodine solution. Every 30 seconds, a sample of starch will be removed from a boiling tube and added to one of these to see whether there's actually any starch left in that boiling tube, or whether the amylase has done its job and digested the starch. I have 5 tubes ready, each containing a mixture of starch solution and a pH buffer, which will keep the pH constant in that tube. So we have pH 2, 5, 7, 9 and 12. I also have some 1% amylase, and both the starch and the enzyme are going to go into a warm water bath at 37 degrees C to come up to temperature. Now that they're at temperature, I'm ready to add amylase to my first boiling tube. A fine syringe is a good choice of equipment because it has a very high degree of precision, so I can be certain that I'm adding exactly the same volume of amylase to each tube. More or less simultaneously, I'm going to add that amylase, start the stop clock, and then also remove my first sample, which really I could have done before adding the amylase, and I probably should, but never mind now. So we can see that at the moment there's still starch in that sample, and therefore the iodine has turned that blue-black colour. After 30 seconds, I remove my next sample, and you can see that the colour is a bit lighter, but there is still some starch left. And after a minute, all of the starch has been digested. So I can stop the stop clock and write down that at pH 7, the time taken for all of the starch to be digested was a minute. I'm now going to repeat the same investigation, but this time using the solution that has pH buffer 2. Again, I'm going to add the amylase, start the stop clock, and take my first sample. And again, because there's starch present, we see that dark blue-black colour from the iodine. Now I'm expecting that this time this will take longer for the starch to all be digested, because I know that depending on the kind of starch I'm using, the optimum pH is likely to be near to 7, which is what I would expect for salivary amylase in the mouth, or a more alkaline pH if it's amylase from the small intestine. So a pH of 2, which is very acidic, is very unlikely to be good for the amylase, and therefore it's quite likely to denature the amylase so that it can't digest the starch at all. If the amylase is still able to work, it's going to be working very slowly because that low pH is going to have affected the active site of the enzyme, and so it won't be able to bind properly with its substrate, and it just won't be able to work as effectively. So I continue to remove these samples every 30 seconds, and in between I'm placing the tube back in that hot water bath so that we're trying as far as possible to maintain an optimum temperature for the enzyme, because if it was too cold, then the enzyme would also work more slowly. So now we can see after 5.5 minutes, there's still starch in the tube. The enzyme hasn't been able to break it down, and so it's fairly safe to say that that enzyme has been denatured. The required practicals present a prime opportunity for the exam board to give you an extended response question in which you're asked to lay out the method for the investigation. Remember, it's absolutely fine to give your answer in the form of bullet points or a numbered list for the method, and I would really encourage you to do that because you want your thinking to be as clear as possible and for it to be in a really logical order. And so writing in bullet points is going to help you to make sure that you're laying everything out really nicely and not missing out any detail or waffling. So pause the video here and see if you can write out a method for this investigation. The first part of the investigation is preparation, so we need to sort out the tubes of starch and buffer solution and also the spotting tile with the iodine in it. So your first 2 marks are for preparing the tubes with the starch solution and also the spotting tile. Then we need to place those into a warm water bath to come up to temperature, and then we're going to add a set volume of amylase to your first tube of starch solution. You don't need to specify a volume, but often it's easier to do that than to just explain that when you repeat the investigation, you're going to use the same volume. As soon as you do that, you start the stop clock, and then at 30-second intervals, you remove a sample of the solution and add it to a well of the spotting tile. And remember, those wells already contain iodine. Now, if there is starch there, we expect a colour change, but if there isn't starch there, we don't expect a colour change. So when the iodine stops changing colour, this will show that all of the starch has been digested and it's time to stop the clock and write down the time. Then you're going to repeat the investigation using the same volume and concentration of starch and amylase, but using a different pH buffer, because we can't determine what the impact of different pH is if we don't test it at different pHs. The pH with the shortest time is the one that is closest to the optimum pH of the enzyme. So in the two that we've looked at earlier in this video, pH 7 was closer to the optimum pH than pH 2 because it took far less time for all of the starch to be digested. The final thing we need to consider is how to interpret the data produced by this investigation and how to improve the investigation. So you might have a results table that looks something like this. And what this shows us is that for pH 2 and pH 12, after 4 minutes, there is still starch present. And that's a pretty clear indication that those extreme pHs have denatured the enzyme, or at least if they haven't, then they're making the enzyme work very slowly indeed. For the pH 9 solution, we can see that it took over 3 minutes for all of the starch to be digested. So the enzyme is working, but it's working much more slowly because the pH is not the optimum. And then pH 5, the enzyme is working a little bit faster, and at pH 7, the enzyme is working very quickly indeed. So out of those 5 pHs, we would pick pH 7 as the closest to the optimum for that amylase. But we actually can't say that the optimum pH is exactly 7 because we haven't tested pH 6 and pH 8, and actually you can have pHs as decimals as well. So in order to improve the investigation and give us a more conclusive result, what we would want to do is to test more pHs. But we don't just want to test any old more pHs. It wouldn't do us any good to test pH 10 because we already know that pH 9 and 12 are much slower than pH 7. So what we want to do is to test more pHs, but to test them within a narrow band that is near to that optimum pH. So we want to test several more pHs from pH 5 up to pH 9. So for instance, we might test pH 7.2 and pH 7.4. And that would mean that we could be more precise in identifying the optimum pH for this amylase enzyme. So that's it for the amylase required practical, and I hope that you found this a useful introductory video. If you did find it useful, then let me know in the comments below, and don't forget to like and subscribe for more GCSE Biology content coming soon.